Okay, so it's our great pleasure to welcome Dr. Brangwynne for his award lecture. So as I very briefly sort of mentioned, very kindly yesterday in the Early Career Researchers Networking event, Professor Brangwynne gave a very stimulating and also thoughtful talk entitled Adventures in Science for Early Career Scientists in Kyoto University, introducing and presenting his careers as a scientist so far, and also insights on what might be key to ask a good question in science, and how you might choose a good pathway to become an independent PI and continue to be a good scientist. I think it was such a privilege for those who attended the event to listen to his career pathway so far and many of his original ideas and also passions for science. And accordingly, there were so many questions from attendees and young scientists, and also following discussions, interactions among the participants and Professor Brangwynne were also, I think, very fruitful. And I myself learned a lot from his talk. So we sincerely thank Professor Brangwynne for taking his time and effort to come over to Kyoto for this event and the award ceremony today. So here, Professor Brangwynne will give us his award lecture on living droplets, liquid phase separation in cell physiology and disease. So Professor Brangwynne, please.
Okay. Okay, Professor Saitou, thank you so much. I've prepared some formal remarks that I'll make later to express my gratitude, but for now I'll just say this is really an incredible honor, and certainly to be the inaugural recipient of this award I think is very special. So I thank you so much. This is really quite an incredible honor. I'm going to go through some of the history here in the work and how we got to developing these ideas. And through this discussion, I also want to touch a little bit upon my personal trajectory. As Professor Saitou said, we spoke a little bit yesterday with the students about that. That was really very fun for me. And some of those themes will continue in today's lecture. I think I want to start out by saying what I view as the fundamental problem in cell biology today, which is the question of how the components of living cells come together, how they assemble or really self-assemble into cells and tissues and organisms. We're at the point now in modern cell biology that we essentially know all of the parts. We have proteomics, transcriptomics, metabolomics, lipidomics. We basically know everything that's in cells. So the question is not so much what are the parts, but how do they come together to form the structure, to form, let's say, a living cell. So this is actually the car I drive, nice Japanese vehicle, Subaru. And so, I think this gives you a sense for the problem that I'm describing, the state of cell biology right now. But it's also very misleading because this is vastly more simple than living systems. So the number of components, the complexity of the components in this example of a vehicle is orders and orders of magnitude, many times. This example is less complex than living cells. So this is the challenge we face, how to think about it, how to think about this self-assembly problem that, living cells. Now, this is a nice sculpture made from Legos. This is a little bit of an exaggeration, but only a little. Say this is the textbook view of biology. And the reason I say that is because we have this idea that individual components, if we study textbooks on biology, the components snap together like Lego pieces, or maybe something like a mechanical watch or an automobile. They, very specific pieces that fit together in very specific ways. And so this I think has been misleading as well in terms of what's actually happening in living cells. And the reason I say that is because the components, this is simulations of proteins in living cells, one of the key building blocks of life. The components are actually not individual structures with a static shape, but they're very dynamic. So even a quote, well-folded protein in a particular conformation, it's fluctuating around quite a lot. So it's sort of, if it's a Lego, it's a shape-shifting one. So it's changing its shape quite a lot. It's also the case that many proteins are not particularly well-folded like these structures, but have regions of significant conformational heterogeneity, what are referred to as intrinsically disordered regions or intrinsically disordered proteins. So the components are variable and dynamic and wiggling around quite a lot. The other part of this complexity is that they're also wiggling around because of Brownian motion, the fundamental fluctuating nature of energy at the microscopic scale. So this is essentially the kinetic energy of molecules that is directly connected to temperature. So for all these reasons and the number of components and their fluctuating nature, this is a huge problem. This is, in other words, living cells are nothing like mechanical machines that we're used to thinking about. So I think that how molecules in this dynamic soup, the fluctuating molecules, how they self-assemble into these structures is something that I started thinking about as an undergraduate. And I'll tell you just a little bit about this history because I think it's interesting. So I majored in material science and engineering. As was said, I learned what that was from a car ride home from a party in high school. And so I ended up going to Carnegie Mellon University and studying material science and engineering and learned about these processes of crystallization where you have individual atoms. You could heat them up and you'll have a molten state of this, in this case, non-living matter, right? A molten state. And as it cools down, this is an ingot that has crystallized, you get solidification where the atoms snap together in register, somehow self-assembling into this particular structure. So nobody has told the atoms where to go. They somehow know to go to the right place to form this. Now, around this time, I also took an introductory biology course and met a graduate student named Vivek Abraham and his mentor, faculty member, Fred Lanny. And Vivek showed me movies like this of living cells. So this is a neutrophil, an immune cell, that is crawling around and it's chasing down this little bacterium. And it's literally hunting this bacterium and trying to find it and destroy it because it could be the cause of an infection that would be problematic for the human. So it's finally triumphantly captured this bacterium. So this is amazing. And as looking at this as I was taking courses in material science engineering, it's very obvious this is an amazing material. If we're going to think about this as a material, it's an amazing one. And moreover, through the biology class, I learned that it's these individual molecules that somehow are encoding for this sort of behavior. So much like, albeit much more complex, much like the atoms snapping together into these simpler non-living matter structures, individual biomolecules are somehow forming these incredibly dynamic and interesting kinds of materials in life. So this is something that I was very much interested in. I actually had this is also something I like to tell the students about. I had a very non-linear path through my studies. I actually dropped out of college for a year. I think I always envisioned going back, but I left college for various reasons. I can talk about that later. And I went to work at Harvard Medical School. I was inspired by some of the work of Don Ingber, a well-known professor at Harvard, and Kit Parker, who at the time was a postdoctoral fellow at Harvard, is now also a faculty member at Harvard. And I became interested in these problems of cellular organization. We were using micro patterns to put individual cells, and it made a discovery that cells can actually coherently move and break symmetry into these kinds of little mini tissues. This was where I think it really, for me, became clear that biology was amazing, and I wanted to study biology, and I was trying to figure out how to make progress in that emerging passion from the perspective of materials science and engineering. Now, the materials we were studying at Carnegie Mellon, in my coursework there, were more like metals, steel, semiconductors, copper alloys. But it turns out that the materials, the non-living materials that are most like living matter, most like cells and tissues, are in this category. And so I was introduced to Dave Waits, who became my PhD advisor from my time at Harvard Medical School. And so I joined his lab for my PhD. He's a pioneer in what's called soft condensed matter physics, or the physics of squishy materials. And so many of these are familiar from everyday experience. And these kinds of materials, even though they're non-living, they're very interesting in that the molecules self-assemble into really interesting types of structures and interesting materials. I'll just give you this example, foam. A foam is something like 95% gas and 5% liquid, and yet it makes a soft solid. So that's kind of very interesting. And in fact, researchers going back to the 1800s have been wondering whether tissues have foam-like behavior. So there's lots of inspiration from these materials to think about cells and tissues. So, from that perspective, I became interested more and more in the organization inside of living cells, the substructures within a cell. And so this is a kind of a classic picture of a living cell, a eukaryotic cell, the kinds of cells that are in our bodies. And you see that there are all these substructures. These are referred to as organelles. And that name comes from the fact that these are to the cell what our organs, heart, liver, spleen, are to our bodies. So these are like the little organs of the cell. And what we're taught in high school biology or introductory biology is that these are like little soap bubbles. They're little membrane-bound vesicles. And that's a clean picture because what it suggests is that there's a boundary, a wall that's basically keeping things in and distinguishing outside from inside. What is true, though, and we started to realize this, is that this is probably just the tip of the iceberg, as we like to say. There are many bodies inside living cells that do not have membranes, and instead we refer to them as membrane-less organelles or biomolecular condensates. For reasons that I'll say more about. So these are structures that are found throughout many different cells and many different locations. These are examples in the cytoplasm. So these germline P granules, I'll say a little bit more about in the embryo of C. elegans. This is processing bodies in living cells that are involved in RNA turnover and decay. There are various signaling assemblies like these structures. And then inside the nucleus, within the nucleus of living cells, there is substructures that are, again, have no membrane boundary. These are examples include nuclear speckles, nucleoli, which I'll tell you more about, and these beautiful structures called Cajal bodies. So again, these are large-scale organelles, but ones that don't have membranes. And I think the mystery when we started thinking about this was that these kinds of structures are comprised of many thousands or millions of molecules that somehow form a coherent structure in the absence of a membrane wall or a boundary. So the question is, what is holding these molecules together and why do they form these kinds of structures? So this was the state of things when I joined the laboratory of Tony Hyman at the Max Planck Institute for Cell Biology and Genetics in Dresden, Germany. We were very fortunate to be able to collaborate with Christian Ekman, another faculty member there, and Frank Ulischer, who is a theoretical collaborator that we worked with. This is the problem I became interested in at the time. This is the embryo of C. elegans. So this worm, which is a model organism, C. elegans. And you can see that there are these green structures. These are the P granules. So this is this type of membrane-less organelle that is found and distributed throughout the cytoplasm of the embryo. Over the course of about 10 minutes, as this embryo sets up to undergo the very first cell division, these P granules all segregate to the posterior, to the right side of the embryo. Now this is important because when these embryos divide in two, the very first cell division, the cell that forms over here will contain the P granules, and the cell that forms over on this side will have no P granules. The cell that forms over here with the P granules is the first progenitor germ cell and will go on to form the reproductive organ in the worm. And so the idea is that these P granules are somehow specifying the type of cell that's going to form. They're protecting the cell from undergoing differentiation. And the question we were asking was just, how do these P granules get to the right side? How do they know to get there? And without going into a lot of detail about this study, in the course of this research, what we realized was that these P granules are actually liquid droplets. So this is now a movie of a different place in the C. elegans worm, where this green material here is now shown just in white. It's all kind of coalesced. And what we've done is basically squashed the tissue. We have just kind of applied pressure, and that induces shear stresses and fluid flow. And that causes these P granules to drip and flow and coalesce with one another. This looks a lot like oil and water mixtures, or if you've ever seen like lava lamps, that kind of thing. So what this says is that these P granules, the green structures here, are really little liquid droplets that contain the biomolecules, that contain RNA and protein. And so together with, again, a lot of other data that I won't go into, this led to this picture wherein the segregation of these P granules, the way that they move over to the right side of the embryo, is a kind of phase transition, what we refer to as liquid-liquid phase separation. It's called liquid-liquid phase separation because the structures that form, these P granules in this case, are liquid droplets, and they form in a surrounding sea of another kind of liquid. So what's happening here is that the droplet phase is disfavored here. In other words, the droplets tend to dissolve on that side of the embryo, and they tend to condense on this side of the embryo. So this is something that we were very excited about, and we suspected that it could be a principle that was more general. As I'll say, maybe we didn't realize quite how general it was going to be. The idea shown schematically here is that the molecules initially, they're distributed throughout the cytoplasm here, like soluble molecules floating around. But under changes in the conditions, it could be chemical changes, so post-translational modifications to proteins, the transcription of RNA changes, epigenetic changes to DNA. There can be thermodynamic free energy changes to the system, so that the total free energy of the system that cause these molecules to condense into a droplet state. And what's remarkable, I think, is that these droplets, in the same way that water droplets can form and be stable even when there's not a boundary or a membrane or an enclosure, as shown schematically in this movie, the molecules can form a coherent structure without a wall, without a membrane. So this is something that, again, is well known in the physics of non-living matter and fields like condensed matter physics, soft condensed matter physics, chemical engineering, and seems to be relevant in many places in biology. These are some examples. In the years since, we've done a lot of work with this in purifying proteins and looking at them in vitro in a test tube. And what one can see here is that these droplets readily form under many conditions in a test tube. So proteins want to phase separate. This thermodynamic driving forces can be recapitulated very easily in a test tube with proteins that form these droplets. So what we now understand, I think that initially we were thinking about them as purely as reaction crucibles, as is often considered for traditional organelles, where you imagine that the concentrations of molecules are much higher, and so you're increasing reaction rates, for example. We now also understand that in some cases, these condensates can do the opposite. They can sequester molecules and actually turn down the activity, the signaling activity. Condensates can also do other sorts of things, and I'll say more about that, but they can function as organization hubs to control the three-dimensional organization of other structures within living cells, for example, DNA and nucleic acids. So this is something that I think is very interesting. Now, there's a long history of thinking about aggregates, irreversible aggregation into gel-like structures or amyloid-like fibers in literature. As was mentioned in the introduction, and is a place that's very special to me is the Marine Biological Laboratory. Jacques Lerbe, this is a paper by Jacques Lerbe from 1921 and his son, Robert Lerbe. Jacques Lerbe is the founder of the physiology course where I spend summers, and he was studying the aggregation of proteins like casein and gelatin, so this has a long history. And more recently, there's been a lot of interest in the idea of amyloid-like fibers and gel-like states forming for many different types of proteins, including these nuclear pore complex. Often what people in this area will see is purified proteins will form a gel. This is actually in the shape of a test tube because the system basically gels and forms something like Jell-O or gelatin across the test tube. That's different. We actually understand something about the connections between these now, but that's different from the phase separation process where these droplets actually separate out from the surrounding liquid in this liquid-liquid phase separation process. And generally, although there are exceptions, we think about the liquid states as being physiological states that promote reactivity, and these solid-like states often being more associated with pathologies, for example, in neurodegenerative disease. So liquids are very familiar to all of us. I always say, I think everyone in this audience has interacted with a liquid today, probably in the last hour. Maybe you're drinking water as we speak. So liquids and the idea of the liquid state of matter is very common. There's actually a lot of interesting physics still being studied about liquids in non-living matter, and these kinds of emulsions and liquid behavior. It's very important for many industries and cosmetics, et cetera. So this is in non-living matter, and I think what we've discovered is that liquid states of matter are also ubiquitous in living cells. Essentially, we're at the point now where any area of biology that you can name, something related to endoplasmic reticulum or endocytosis or transcriptional activity, you'll find hundreds of papers about condensates and the role that condensates play in that process. So this spans cell biology. You'll have whole issues of plant cell on condensates in plant organization. Many connections with human disease. I'll say more about this later. And many applications where we and others are interested in engineering condensates for doing specific things in cells to forward engineer and control cellular activity and organization. So this has become very exciting over the last 17 years or so since our 2009 paper came out. So I'm going to explain a little bit about this idea of multivalency, which is a key concept in the area. So imagine I have particles, billiard balls or tennis ball or whatever you like, and imagine that they're inert, so they don't stick to anything else except for, we're going to paint a little sticky patch here, a little bit of glue. We'll paint in this red spot. Now, if I then take a bunch of these particles and mix them up, some of them will find each other in the right orientation and they'll stick together. And that's basically the end of the story. There's nothing else that can happen. So for a valence of 1, in other words, 1 sticky patch, I have a dimer, basically a 0-dimensional object, just a little point. But you can see what starts to happen if I paint 2 sticky patches, so valence of 2. Now I can start to get filaments that form. I can get 1-dimensional objects that do something slightly more interesting. And you can see where this is going. If I paint 3 patches, I can now get connected sheets, and higher valence will give me space-spanning connectivity across the system. So if I'm going to form a structure, like a 3-dimensional structure with many components, they're all interacting, I need to have a high degree of valence of the molecules. A single molecule that dimerizes won't quite do it. This idea has been elucidated quite nicely over the years, and this is an early, very important paper by Mike Rosen's group, where he and his colleagues showed that repetitive domains of proteins, which are very common in living cells, so that these magenta squares interact with the SH3 sites. And so when you purify proteins like this and mix them together, they will form condensates in a test tube. So this is something that seems to be very common in living cells. There are also, I already mentioned, proteins often have these disordered regions, what we call conformationally heterogeneous regions or intrinsically disordered regions of proteins. These are elongated polymers, and almost by definition, they are multivalent, right? Polymers, many subunits, so that there are many places where you can interact with this chain as a multivalent region. So these are the ingredients to build 3-dimensional connected networks of interactions dynamically in living cells. So one thing that's interesting, though, is that many proteins, actually, and many proteins found in these condensates, they have both IDRs, these disordered regions, and oligomerization domains. And I'll explain that a little bit. This is a protein that we're very interested in, nucleophosmid. It's a key component of one of the structures that we study called the nucleolus, which I will tell you more about in a few slides. This has an oligomerization domain, so this orange domain, it promotes pentamers, so 5 connected interactions, as you can see here. And then it has this disordered region in the middle, and then there's a substrate binding domain or an RNA binding domain in RBD. So this is common and essentially gives you this sort of high valence of interactions, which is mediated as well in this case by the presence of RNA in these systems. So many proteins that are key components of condensates. Here are the names of the proteins, and here are the corresponding condensates that they help assemble, have such an architecture. So an oligomerization domain like this one, a disordered region, and then some kind of labeled here substrate binding domain. Could be a binding RNA, maybe binding some epigenetic modification or something else. So this is interesting. I want to draw your attention to this. What's really happening with this oligomerization? We said that it gives you this higher degree of multivalency. You can also understand that from the perspective of really fundamental concepts in polymer physics. And one of the things that we talk about a lot in this field is the idea of what's called Flory-Huggins polymer theory, or regular solution theory is kind of the simpler version of it. But the basic idea is this is an expression for the free energy normalized by the thermal energy scale KT. And these two terms, you don't have to worry about the details, but these two terms are basically speaking to the entropy of the system, so which wants things to be mixed, doesn't want droplets of high concentration surrounded by a lower concentration phase. This is the term that says for some favorable interactions, you might want to form droplets, although it would be energetically costly with respect to entropy, entropically costly. The 1/n we think about as the degree of oligomerization. So the higher the degree of oligomerization, the less important, at least that entropy term is. So this is a little bit abstract, but I'm going to show you in a specific example how we can use these concepts to engineer condensates in living cells and understand how that really works. So this is a set of technologies, really a whole suite of technologies we've developed over the years that uses optogenetics, which is the use of light, externally applied light, to control molecular interactions and to control the oligomerization state of molecules in cells. This is some work that started from a really amazing former postdoc, Youngdae Shin, who's now faculty at Seoul National University, Dan Braha, now faculty at Technion University, and my colleague at Princeton, Jared Tocher. So all of these technologies basically do the same thing. They use light to control oligomerization, and this allows us to write droplets into living cells. This is the nucleus of a living cell. At the beginning of that movie, we shined light. It was otherwise pretty homogeneous except for these nucleoli, and these droplets nucleate. This is a little bit like a cold, glass of lemonade or iced tea or something on a hot, humid day where you're getting droplets that are condensing, except these are biomolecular condensates condensing in the nucleus of a living cell. We can also do this and control how these droplets interact with genomic elements inside of the cell. I'll say more about that in a moment. And because it's light controlled, we can actually steer the laser and write droplets, create droplets at specific subregions in the cell. And for example, draw smiley face of droplets in cells. So again, this is sort of fun, but also I think somewhat remarkable or crazy that we can actually write, draw patterns of biomolecular condensates in living cells. So we've been using these technologies for a lot of different things. What's fun for me is that it takes me back to the materials science as a first-year undergraduate. We're pouring these molten alloys. We're doing something similar here, except we're changing these states of matter of biomolecules in a living cell by controlling the oligomerization state with light. So that's, I think, again, something that we're very excited about. To take you back again through the adventure back to Carnegie Mellon, in materials science and many of these related areas of physics and applied physics, we have this idea of phase diagrams. These are essentially fingerprints that describe the conditions under which matter self-organizes. So this axis is the weight % carbon, so some concentration axis. This is a temperature axis. And for a given point in this phase diagram, it says whether you have these kinds of structures that form or these kinds, or whether at high temperature it's a liquid. This is actually notes from a class that I took called Phase Relations at Carnegie Mellon in the late 1990s. I think it's no exaggeration to say that the ability to understand the phase behavior of non-living matter has been foundational to our modern world. So, everyone has a computer in their pocket right now, I suspect, and that's all been enabled by the semiconductor industry, by the ability to harness the phase behavior of silicon to control and compute, right? So that's really what this is all about. And so a lot of us, have this vision in the condensate field. What if we can actually do something like this for living states of matter to control and engineer in living cells? So we're able to do really the first condensate phase diagram in living cells using these optogenetic approaches. And so this, again, is something very exciting for me to see. This axis is the oligomerization state. So again, whether it's dimer or a 5-mer, like those molecules we saw. This axis is the concentration, basically the concentration of those molecules. The open red, the solid red circles you see here are cells at these conditions, and those ones phase separate. So if we shine light on that cell, we get droplets that form. These open red circles are cells at those conditions where we do not get phase separation. So you shine light, nothing happens. The cells that do phase separate, you'll get a dilute phase. So these blue triangles is the concentration that forms of the dilute surrounding phase. And the blue diamonds here on this side are the concentration of the condensate that forms. Now, if you've ever taken any undergraduate physical chemistry class or a polymer physics class, you might recognize these kinds of shapes. This is from the famous polymer physics book by Colby and Rubenstein. You have this kind of upside down parabola shape. It says, you get phase separation when you're in this region of the phase diagram and no phase separation in this region. This axis here is again a concentration, some volume fraction. But this axis is actually a temperature axis. And so we think this is really interesting because here I have a oligomerization state, and I told you that was controlling entropy. It's basically dictating how important the entropic contribution is. And we think about temperature, from fundamental thermodynamics as also kind of modulating the entropic contribution. So we think about T delta S terms, if that means anything to you. So this is basically, oligomerization here is essentially an entropic knob that we're turning in living cells, and we think that natively this is happening by cells. The other thing that I think is really interesting and is different in this space compared to traditional polymer physics, where most of the polymers they're homopolymers. You have one repeated subunit. In proteins, you have 20 different amino acids, naturally forming amino acids. So that means you have 20 to the n, where n is the number of amino acids in a chain, which typically is in the hundreds, 100 plus. So the combinatorial complexity is quickly astronomical. The number of different chains, the flavor of the chains is vast very quickly. And the question is, what sequences give what phase behavior? In other words, if I tell you a particular sequence, can we say what the phase behavior is? This is a great challenge that has been solved in an analogous way in the AlphaFold problem, but for this higher order organization, it has not been solved. We've been doing work in this space, though, to map these phase diagrams. This is like a kind of a temperature axis, this is a concentration axis, and we can measure phase diagrams for these different related sequences. So this is, again, a program that is very much underway in this field. Another thing that I think is very interesting in making these connections between what is happening in living cells and the kinds of physical phenomena, the kind of physics that's happening in non-living matter and traditional material science, is this idea of classical nucleation theory. And I would say, again, this is something that takes me back to early studies in materials. This is a really simple idea, but it's interesting. We can think about this again as a kind of a free energy expression. This negative sign is good, right? So that means this is lowering the free energy. This, you see, has a volume-like term, and this is a volumetric free energy. So this is a term that says, yes, we're going to lower the free energy by phase separating. This is a positive term, and that means it's going to increase the energy. And you see that this has got an area dependence, and there's this gamma term, which we're going to see again, so I want to flag it already. This is what we refer to as the interfacial tension. And the basic idea is that if I'm going to form droplets, if I'm going to do this phase separation process, starting from a homogeneous solution, and then now I get droplets, those droplets have an interface, and those interfaces that where the droplet meets the surrounding phase is a high energy region. So this interfacial tension is basically an energy per unit area that is a penalty you have to pay. And because small droplets have a large area compared to the volume, we think about surface to volume ratios going like 1 over the size of the droplet, this basically wins in the beginning. If you make a really small droplet, this wins, and so the energy goes up. Only when you have a larger droplet can you get on the other side of it and actually favorably decrease the free energy of the system. This is exactly why if you have something like carbonated water, seltzer water, soda water, you often have bubbles of carbon dioxide that nucleate in little cracks in the glass, little micro defects in the glass. It's because this is a place where you can partially overcome this energy cost by forming the bubbles here. Now this is a very sensitive and well-controlled experiment that I did in a hotel room in Germany with a glass with carbonated water. But it's actually very simple. It's just taken with my iPhone. This is a nice movie. You can see the nucleation of these carbon dioxide bubbles. Again, it's exactly this, and it's exactly for this reason. So these are all classic ideas, and the question we were wondering is this relevant for living cells? Now, this is fun to share as well, because this is work done by former postdoc Shunsuke Shimobayashi, who's now a faculty member here at Kyoto University. And Shun was very excited to explore these ideas. And so this is a cell. I'm just going to give you a little bit of flavor. This is a really beautiful work. You can read more about it. But these are cells where what Shun has done is he's engineered these little seeds shown in green. So these are really not condensates. They're little seeds of disordered regions of proteins. And the question is, when he shines light on the cell to induce phase separation of this same type of protein, FUS, where do those condensates form? So do they form everywhere, or are they specifically forming at these seeds? And it turns out when the cell is only moderately supersaturated, so in other words, it's just a little bit over the phase boundary, what Shun saw was that these red condensates tend to form on the seeds. And you can see that here in this blow up. So this is much like what we showed with the seltzer water. They're nucleating at these specific sites because the free energy can be lowered there. But if you take a system, and this was enabled because Shun had very carefully mapped the phase diagram of these systems, if you take the system and push it harder, this is a little bit like shaking up the can, maybe then handing it to your friend. That's not a nice thing to do. But then the bubbles go everywhere, right? And so that's basically what happens here. Strong supersaturation, you get these condensates form everywhere. They don't care about the seeds anymore. So this is, again, a lot of beautiful quantitative work in connecting with classical nucleation theory and deviations from it that Shun elucidated in this paper. So all of that was about this idea of interfacial tension, that gamma term that I highlighted. So I describe that as an energy cost per unit area. So it has units of energy per area. But if I think about an energy, you remember from freshman physics, an energy is like a force times a length. So I can also look at the dimensions of gamma as a force per length. If this is force times length over length squared, this is force over length. So this is sort of a little bit of starting to think about interfacial tension as a way of generating force. And these kinds of forces are referred to as capillary forces. I was told that we have Cheerios in Japan. I have not confirmed that, but I believe that's true. Anyways, this is a breakfast cereal. These little circles, they float on the milk, right? And they're floating around the milk, and you'd say they don't care about each other. But when they get close enough, they zip together. And now they tend to stick together. So that is entirely a capillary effect, in that the Cheerios are deforming the surface of the milk, and if they share the burden by getting close to one another, then they lower the free energy of the system. So that generates a force that actually pulls the Cheerios together. There are examples of such capillary forces in the natural world. For example, oh, this is, sorry, I didn't even show the movie, sorry. These are the two Cheerios, they're close enough, and then zip. That was a pretty thrilling movie. You're welcome. There are examples of these kinds of effects in the natural world. This is a meniscus climbing bug. This is a bug that deforms itself to generate forces mediated by that interfacial tension of the water in this case. So, given that these biomolecular condensates, liquid states of biomolecular matter, we now understand are ubiquitous. They're everywhere inside living cells. We wondered, maybe they can actually function as sites of force generation. And so we've been thinking about that, and this is just a review article we wrote to kind of talking about these ideas that condensates could be sites of force generation, where forces could actually be applied. If you do a back of the envelope calculation, the scale of the forces that could come from these sorts of effects are comparable to the molecular motors, which are the classic force-generating units in living cells. So we want to test these ideas. Again, you'll see a lot of what we do is using, sometimes people refer to as biomimetic systems, these engineered systems harnessing what's happening in living cells to see whether this is possible. So this is a system, this is building from Shun's work, where we learned a lot about nucleation and how to control where condensates form and how to engineer sites where they bind to specific regions of the DNA. This is the nucleus of a living cell. These here, again, these are not condensates. These are these engineered landing sites, these seeds that are, we've set this up so that these are at specific places in the genome, specific genomic loci. And that's really the power of this. We can try to ask these questions about force generation on specific regions of the DNA. And so what I'm going to show you here is we nucleate. This is, I don't have the pictures shown here, but this is work from former postdoc Amy Strom and Yoonji Kim. Really a beautiful, beautiful study. What they've done here is to nucleate droplets, nucleate these condensates on the landing sites and get them to fuse with one another. And you'll see this now. Here comes the green condensate, starts to grow, and then they touch, and now we get a single droplet. We then turn the light off to shrink it, and as it's shrinking, it actually pulls these genomic elements together. So this is, if any of this was unclear, it's basically like the Cheerios effect, right? Except this is with biomolecules that we're controlling with light in living cells, and we're using these capillary forces, using these effects to pull on specific elements of the DNA, to pull DNA together. There's a lot in here that I would encourage you to check out if you're interested in this, but we use this as a way to actually measure the mechanics of the genome, so the stiffness of these genomic elements in a way that was not possible before. So a lot of what I've shown you is are examples of condensates in the nucleus of cells. Also showed examples, including in the very beginning, P granules, stress granules. There are other things in the cytoplasm outside of the nucleus, but a lot of what we've been talking about is inside the nucleus. So remember that the nucleus is this amazing place where about 2 meters of DNA in every one of your cells is compacted into this very tiny structure, which is where all of the genetic information is stored. It's quite remarkable. So condensates are truly everywhere inside the nucleus, subcompartments. These are some of papers from our lab. I'll tell you more about this structure, the nucleolus, which is just an amazing structure. that controls the production of ribosomes. There's a lot of interest in RNA polymerase II, sort of the background household transcriptional activity inside of cells, for which there's intense interest because in many cases the activity inside these kinds of condensates is connected to diseases, especially cancer. A lot of interest in this area. And then this is about activation of the genome. There's many structures, many condensates seem to suppress the activity of the genome in ways that we're still figuring out how that all works. But silencing the activity of the genome. And there's a lot of work now on the intrinsic capacity of the genome to itself phase separate, DNA with associated proteins to phase separate into these kinds of condensates. So this is a very interesting place to be asking questions about these liquid states of biomolecular matter and how they're interacting with the DNA and how they're controlling genomic activity and expression in cell physiology and disease. The structure that I want to draw your attention to, we've mentioned once or twice, is the nucleolus. So this is a field of cells, single cell here, single cell here, single cell here. This is the nucleus of the cell, nucleus of this cell. And then the nucleoli are these substructures within the nucleus. These are really, really interesting. This is the largest of the membraneless condensates in cells. It actually has a multi-phase layered architecture I'll say more about. But the whole, I'd say the point of this structure is to help build the ribosome. And ribosomes are the machine that does protein translation. So we think about DNA making RNA, and that RNA being decoded into proteins through the process of translation. All of that is what the ribosome does. The ribosome takes the RNA code and turns it into the protein hardware. So this is an amazing machine. It is comprised of 4 different RNA components. The RNA is actually where the business happens. That's the catalytic activity of the ribosome is really RNA-based. But it has about 80 different proteins in humans, and it forms these 2 subunits, the large subunit and the small subunit. A typical cell in your body right now, let's reflect on this for a second. A typical cell in your body, and certainly a growing cell, is making about 1,000 ribosomes every minute. So during this talk, you've made a lot of ribosomes. That process also is upregulated when cells become hyperactive in diseases. So we're very interested in how all this works. The RNA components that I mentioned are at the catalytic core are transcribed in the nucleolus, and they're subject to hundreds of modifications, chemical modifications, cleavage steps to form this machine. This is really no other word I know of other than mind-blowing, sort of crazy. What happens is, I mentioned 4 different RNAs are in this. 3 of the 4 RNAs are made on one single transcript in the nucleolus. These have different labels, the 18S, the 5.8S, and the 28S. Those are just names, don't worry about it, but these have to get cleaved. That's why the little scissors here. They have to get cut from this single transcript to form the individual RNAs that again, make up the heart of the ribosome. So we've been interested in all of this and how to think about the relationship between this amazing processing and assembly and the liquid multi-phase organization of this structure. Last thing I'll just say on this slide is that these are also just names. We don't have to worry so much, but there's this inner layer we call the FC, this intermediate layer called the DFC, and the outer layer GC. So this is something we're also very interested in. What does it mean to have a multi-layered liquid, and does that even make sense? It's different than what we usually think of with oil and water mixtures. We've been working on this problem actually after the P granule study. I focused my attention on the nucleolus, and we've done a lot of work in different systems to study the nucleolus, and we've shown that it is a multi-phase liquid. In fact, this bottom movie I think is fun to show. I'll play it again. This is in a frog system where you get many nucleoli that form. These are active and alive. But you'll see that they all coalesce, as you'd expect for liquids. Although you'll notice that the green stuff inside, it also coalesces, but more slowly. So it's this idea of a kind of liquids within liquids, kind of like a Russian doll kind of nested structure. We've also been really interested ultimately in thinking about how all of this is connected to the thermodynamics of this process and the non-equilibrium steady state features of ribosome biogenesis. So this is the set of questions that has occupied us for some time now. I want to draw your attention to this study that's a recent paper that was led by Holly Cheng, who's now a graduate student at MIT. She was an undergraduate at Princeton, very talented student. And we had some amazing collaborators, including especially Zheng Shi at Rutgers and his student Juan, and Howard Stone at Princeton, and James Rogman, his student. This is an approach called micropipette aspiration, which is basically like taking a straw. We all, I believe, have the intuition if you take a straw to water, you apply this back pressure, the water comes up. If I do that to honey or some kind of very viscous thing, I'm gonna have to apply a lot more pressure to have it come up into the straw. So it turns out you can actually calibrate that and learn a lot about the material, how viscous it is, whether it's not a simple liquid but has other properties. And so Holly learned how to use this approach that Zheng had developed for purified condensates, and she applied it to these living nucleoli in the frog system and was able to measure their properties. I'll play that movie for you again, just sort of fun. So this is basically applying a back pressure and slurping up the nucleolus and measuring its viscosity. One of the things that I think is quite remarkable is, let's try to see it in this movie, but Holly, if she keeps going here, you'll see this GC material, this outer layer material, gets slurped up, but when the inner layer hits, it sort of gets stuck. So this green DFC, it gets stuck. This is in a time trace. I'll show you that it basically, it's stuck, and then once the green inner material pops through, it then takes off. My children are into bubble tea, and so that's what this is. This is microscopic bubble tea with biomolecular condensates, right? Everything's fine until the thing hits the straw. So these are multi-phase liquids, but the properties of the different substructures, are actually very different, and in some cases can have viscoelasticity, not just viscous behavior, but also elastic behavior. So this, what I won't have time to go into, but what Holly showed is that the stickiness, I'll call it, of that inner layer is connected to the high concentration of RNA that's in those structures. So we've known for a while that when you turn off the transcription, when you turn off the production of the RNA inside the nucleolus, the structure changes. And so this is a movie, this is a image processed montage of many nucleoli from individual cells that have just been put together into this movie. This is taken at the Marine Biological Laboratory by students Carolyn and Thomas. And at the beginning of this movie, they add a drug that stops the production of the RNA. And you see that as that happens, these nucleoli dramatically change their structure. They go from looking like kind of misshapen liquids to something that looks more like a kind of an equilibrium liquid behavior. So we've been trying to understand what that's all about. What is happening in this very complex process of the production of ribosomes, the transcription of this RNA at that's the heart of the ribosome, and this emergent self-assembled structure. So a recent study that is, I think, very exciting we did in the lab was led by Sophie Kinado's former postdoc, who's now a faculty member at Johns Hopkins, together with Lifei Jiang, very close partnership, a grad student in the lab, and our collaborators Danilo Fontaine and Sebastian Klinge. And what Sophie and Lifei developed here was an approach to use next generation sequencing, these amazing technologies to sequence nucleic acids, DNA and RNA. And basically what they did is they took cells that are growing, they're doing their normal thing, and they sprinkled in some nucleotides that are chemically modified, but they get in and everything is otherwise normal, and then they get incorporated into the nascent ribosomal RNA. And then Sophie and Lifei can then pull out that specific RNA and sequence it. And what they developed was a clever way to look at the reads from the RNA, whether they span a junction that's been described in the literature. This is a junction where there has to be a cleavage. The scissors eventually come in and cut here. But if we read across this junction, we know that the cut has not yet happened. If we see a buildup of our reads, it would suggest that these cuts have already happened. And so you can basically develop a metric which is the fraction of the RNA that's been cleaved. What's beautiful about this approach is it can be done in what is called a pulse chase fashion, which is basically saying we can measure this in time. And so this is a way that Sophie and Lifei developed to look at so these are the representation of the individual RNAs, these blocks, and then these cleavage sites, O1 shown here is one of the cleavage sites, and O2, this other cleavage site, mapped using this approach. So the fraction cleaved as a function of the time, chase time, but you could just imagine this being a time measurement. You see, these ones happen very quickly. The cleavages occur pretty quickly. Other cleavage steps that happen downstream of this happen a little bit later. These so-called 1 cleavage site and 2 cleavage site, they happen later, and others occur still later. So this has been very powerful because it uses genomic sequencing to quantitatively measure where these processing steps are happening inside this multi-phase liquid structure. Now, Sophie and Lifei were trying to understand what's the relationship between these cleavage steps, the processing of the RNA, and the emergent structure that's forming around all of this. This is a plasmid system. For technical reasons, it's very difficult to genetically modify or genetically engineer the native ribosomal DNA. So what we used and what Sophie and Lifei developed were these plasmids, so little synthetic ribosomal DNA that could be introduced into cells. And what they found is that these structures, basically a normal nucleolus forms around these sites. You get normal production of RNA. But if they mutate some of the sites where these RNAs that help guide and direct the cleavage machinery to the proper location, such that this cleavage no longer happens, the nucleolus changes structure dramatically. It basically goes inside out. And so this is something that is very interesting to us because we want to understand why these liquids are organized in this particular way. Sophie and Lifei showed that if they add the RNA, the guiding RNA that is complementary to this mutated region, they could rescue and get a normal nucleolus to form. The way we are thinking about this, and this actually is inspired by a really important study that my first graduate student, Marina Farrick, did in 2016. And we've been building from that conceptual framework, but it all comes back to our friend, the interfacial tension. Remember this gamma, which we said was an energy cost per unit area. That interfacial tension or surface tension we've already seen. And it's known that again, from soft matter, chemical engineering, these non-living matter, studies of oil and water mixtures, et cetera, that the the organization of these kinds of multi-phase liquids is dictated by the relative interfacial tensions. So you could have, for example, this phase 3 inside of 2, or 2 inside of 3, or maybe in some cases 2 and 3 don't want to touch at all, or maybe they partially wet each other like this. So this is all mediated by these interfacial tensions. We think that that's what's happening in the nucleolus. This is some nice computational work done by former postdoc Hongbo Zhao, who's now faculty at UCSD. And so this is, not proving that this is what's happening, but it's demonstrating or illustrating what we think is happening. This is the normal organization where the DFC, is inside and the GC is on the outside under normal RNA conditions. But when you turn off this cleavage, we think the interfacial tensions change, and then you get an inversion. So you're basically, going from like a state like this to a state like this. We think that's happening because the interfacial tensions are dependent on the concentration of this processed RNA. We, like many people, are very interested in using broadly defined AI approaches. This is a convolutional neural network approach where we're trying to understand the three-dimensional organization of the nucleolus under various perturbations. I just showed you under a very specific cleavage, the nucleolus inverts. The outside goes in and the inside goes out. So we've been trying to understand that and use a convolutional neural network to quantitatively map that, what I'll call morphospace of the nucleolus under various perturbations. You can imagine genetic perturbations, or in this case, we've done a panel of molecules, bioactive molecules that are known to target RNA. And so Anita Donlich and Troy Comey developed this approach and actually used it to discover a new morphology of the nucleolus that emerges when they inhibit an enzyme called topoisomerase that regulates the processing of this ribosomal RNA. And so with this approach, they were able to map a morphospace and describe the different conditions. This is, in orange, the normal state, or this cap morphology, so-called, when you turn off transcription. Necklace is another morphology described in the literature under a particular perturbation to CDK, one of the cyclin-dependent kinases, and this flower morphology that they've discovered. So this is, I think, something really interesting, and we're pushing on this in various ways. I mentioned already several times that a lot of these ideas are very closely connected to pathologies that very much a concern to humanity, including, I already said, neurodegenerative diseases and cancers. Growing cells have to produce a lot of ribosomes in order to do all the translation that is required. And it's been known for a long time that pathologists, when they look at tissue sections, these so-called H&E stains in tissue histopathology, if you have cells that have nucleoli like this, very large, misshapen nucleoli, this is a bad sign, because what it means is that those cancers are really growing fast. They're really producing lots and lots of ribosomes. So this is more typical of cells, and this would imply a more slow-growing cancer. So we've been wondering about what this means. If cancer cells are upregulating the production of ribosomes, maybe instead of 1,000 per minute, it's 2,000 or something. Can they do that? Can if you just start making more of something like a car factory, and you say, hey, make twice as many cars, that's not easy, right? We got to change a lot to the production line to increase the level. So this is now, I'm going to show you a movie. This is if you've been sleeping until now, this could be a good time to pay attention to this. So I don't know, I actually looked this up. Apparently, I Love Lucy was a popular sitcom in the United States, and I guess it was dubbed into Japanese and was popular. And I guess there was even an episode, The Ricardos Go to Tokyo or something. I didn't look this up. I suspect no one in the audience has any idea what I'm talking about. anyways, this is an I Love Lucy episode. So this is fine, right?
Yeah, we can handle it. Okay.
They're making these chocolates. Everything's fine. Cells making ribosomes. Everything's fine. And the question is, are cancer cells more like this?
Listen, Ethel, I think this.
I think we're fighting a losing game, Ethel. That's what she said. So, in other words, can cells keep up with it? So that's the question that was asked in, I think, this really nice study by Li-Fei Zhang, the same grad student I mentioned before, building from the work that she did with Sophie, together with her co-advisor, Yibin Kang, who's a cancer biologist at Princeton. And the basic question is, when cells are hyperactivated and they're making more ribosomes, what is happening at the level of the efficiency? And we're measuring this. We're going to define efficiency as the number of ribosomes, or the amount of ribosomes you get, divided by how much transcription of the ribosomal RNA that's going in. And so the question then, just like the I Love Lucy episode, is that when you have cancer cells, oncogene hyperactive cells, if I make 3 times as much ribosomal RNA, do I get 3 times as much ribosomes? Or maybe I'm getting more ribosomes, but I'm losing, it's not as efficient. I'm making 3 ribosomal RNAs, I'm only getting 2 ribosomes out. So that's the question. And how to measure it. So this took a lot of technical development, and I'm just going to give you the high level of it. But we do the same technique using the labeled RNA. This is the chemically modified nucleotide that goes in. And you can actually image that RNA, and you see this is the normal cell. This MYC oncogene is low. When Li-Fei turns on the MYC in this cancer cell, you see that it's a lot brighter. So they are making more ribosomal RNA in this case. This is how we measure the protein. So this is out in the cytoplasm, the orange here. This is a halo tag for the aficionados. This is basically measuring how much ribosomes are actually being made out in the cytoplasm. You'll see here, too, we get a brighter signal out in the cytoplasm. But when you actually take the ratio of these signals to measure the efficiency, it turns out that the cancer cell, even though it's making more ribosomes, the efficiency goes down. So I always think about that. Again, it's like the I Love Lucy episode, or if we went to the local Subaru factory and we said, make twice as many cars tomorrow. I mean, they'd make more cars, but there'd be problems, right? You can't just suddenly turn it up. So Li-Fei, sought to build from this earlier work she did with Sophie to, with this technology to map where the ribosomal RNA's processing occurred, to try to figure out if the efficiency goes down in these cancer cells that are trying to make a lot more ribosomes, where is the problem occurring? And so she mapped it out for these normal cells, and then in this case, these malignant CA cells. This is the fraction cleaved of one of these early cleavage sites, O2. And you see that basically it looks like one curve because both of the curves are on top of each other. There's no difference, in other words, for this cleavage site. So she's able to do it for many of the sites. So this is a different cleavage site, site 2. Same thing, the curves are basically on top of each other. But it turns out when you go to these later cleavage sites, you start to see significant deviation. So the malignant cells are shown here. They're not quite keeping up. So it's the I Love Lucy. You're putting more material out, and the enzymatic activity that is required to do this cleavage is just not keeping up. So we think about this as a sort of a bottleneck in the process. It's a place, everything's going along fine, but you get to a position in the assembly line or in the processing sequence where it just can't keep up. So this is interesting because we think about these bottlenecks as potentially vulnerable to therapeutic intervention. If I have therapeutics that hit over here, or at least partially hit, maybe it's not as big of a problem, but if I hit here, it's already a weak spot in the system. And so Li-Fei has been testing these ideas, and this is just to give you a flavor of the kinds of results, but But what she's found is that when she targets knocking down factors that are involved in these late processing steps, she can get shrinkage of the tumors, which I think is very interesting. So again, I'm not trying to say we're curing cancer. This is the answer to to all the problems that come up in cancer. But I think this is a very interesting conceptual framework, and we're trying to advance that to think about therapeutics in new ways. So I told you a lot about things we're doing in the group. I focused a lot on the nucleolus and the production of these ribosomes that ultimately they go out into the cytoplasm and they're doing this translation. They're taking the messenger RNA, like this line here, and they're building proteins from it. This is the heart in a lot of ways, right? This is the heart of biological processes. It's the heart of cancer. Many things are happening that are connected to these processes. We're very interested in that, especially in all of the connections to disease. I mentioned, of course, cancer, aging. I've been telling people every year I become more interested in this problem, and of course, protein aggregation disease. This is devastating as medical miracles. We're living longer and longer, and now we have these emergent problems. The statistics, something like 1 in 9 in the U.S., 1 in 9 U.S. citizens over 65 will develop Alzheimer's. So these are real problems, and we're very interested in them. And we think that the condensate framework we've been developing and these ideas that I've tried to share with you are helpful for thinking about new ways of targeting these diseases. There's a lot of interest in biomolecular condensates in the drug discovery space because there are so many fundamental processes that are linked to condensate organization. Any of those processes that go awry, that something goes wrong in disease, that's a place where we could think about targeting condensates or interacting with condensates to therapeutically address it. So examples, cancer, virus, this is respiratory syncytial virus, again, virology and SARS-CoV-2 and neurodegeneration. There's hundreds and hundreds of papers in each of these areas, and I think it's very interesting. There are drugs going into the clinic now from condensate-based drug campaigns, and that's, I think, very, very exciting. So, I'll try to wrap it up here. I mean, I frame the talk in this. I think this is the fundamental question in cell biology. We know the parts. It's vastly more complex than this, but the problem is really self-assembly. And I think there's many places where fundamental physical concepts from physics and soft matter are relevant here. But I tried to tell you a little bit about the way we're thinking about this and how molecular collections of molecules can self-assemble and self-organize into these liquid states of matter. This is kind of one of the first emergent organizational principles within the cytoplasm and the nucleoplasm of living cells. So there's lots of interesting stuff here. I tried to give you a flavor for it, but this is something I think very interesting. As with the student discussion yesterday, and as you saw in a number of my slides today, I tried to tell you a little bit about my personal journey because I think that it's potentially serves as some inspiration or lessons can be learned for the younger students, the trainees. But for me, this has been very exciting because, again, it all started for me in thinking about the phase transitions, the self-assembly of non-living matter. And we found that a lot of these ideas turned out to be highly relevant for living matter, which maybe shouldn't be surprising because, after all, it's matter. It's the stuff of the physical world. But I think that I hope that my story and the trajectory and some of the things that I've learned along the way and my adventure in kind of bringing these ideas into cell biology has been illustrative and instructive and potentially inspiring for the younger students. As I mentioned, I'm incredibly honored to be here, especially as the inaugural Kyoto University Lectureship Awardee. But I also want to mention, it's really important, I think, that science is a team sport. And this is certainly another message I think for the trainees would be important is that everything we do that's interesting, every paper that I've ever written has many co-authors and is highly collaborative and involves many brains and ideas and creativity. I've been incredibly fortunate to have many mentors, colleagues and trainees. This is an incomplete list. I can't show them all and name them all, but just gives you a sense of the many people that I've been incredibly fortunate to interact with and learn from. I just feel very blessed to be in this profession and to have learned from all these people and really the recognition that I'm so grateful for receiving today from Kyoto University should be viewed as a recognition of this group and the field that has formed around these ideas. I also want to give a special thanks to my family. Again, I can say for the trainees, I think it's important to remind people that scientists are humans. We are not robots. I am extremely fortunate to have an amazing family that has sustained me through, the ups and downs and the trials and tribulations and challenges of doing science. It can be very hard, and I'm just incredibly blessed. I had the opportunity to visit Japan, both Tokyo and Kyoto, in the fall with my wife, Sarah, as part of the Keio Prize activities, and we had an amazing time. Just incredible, and my sister was jealous, so she came this time, and we dress like this around town all the time, but yeah, we've had a really, really fun time, and I think that beautiful thing that you've set up here with the Kyoto University Lectureship Award, and I think it's wonderful to invite family members to join. That I think makes it a very special experience, more so than I think would be the case just coming alone. So that's really nice. I encourage you to continue that tradition going forward. That's been lovely, and Kristen and I have had a wonderful time. We're incredibly grateful for that. This is a picture of the group not long ago, and so I'm happy to have questions and a discussion with you all now. Again, my sincerest gratitude for inviting me here in this tremendous honor. Thank you so much.
Okay, so thank you so much, Professor Brangwynne, for thought-provoking as well as fundamental and also comprehensive talks on your work on the protein condensates. So as this is a very nice opportunity, so we can have a number of questions. So are there any questions or maybe comments from the, please.
Thank you for the really nice and impressive talk. My question is about the droplet the droplet, of course. I want to know about the surface of the droplet. There's a lot of study about the generation of droplet, monitoring a droplet, and check the role of droplets in living cell or outer cell, like a reactor for reactions. But as you told in your talk, maybe the surface, it means the interface between a droplet and others is probably will have a very important role to fission fusion of droplets or generation or disappearance of droplets. But in my understanding, excluding some work you mentioned about the line tension or something, almost all thing refers about the inside or of droplets. So if you have any fun comment of the surface of droplet and how it works, how it composes the orb, or how to say physical characteristics of the surface, if you have any opinion, I'd like to hear from your voice.
Yeah. Okay. Well, thank you. Yeah, I think the question was, really about if I could say more about the interface of the droplet and what's really happening there and how it's organized and all the things it's doing. But we could talk about that quite a lot. I mean, I am fascinated. I've been fascinated by, these condensate interfaces since the very beginning. I'll just say, a couple of things. One is that the surface tensions, the interfacial tensions are low, and that's generally true for macromolecular liquids, including, colloidal liquids. We sometimes talk about work of Lecker-Kircher and others, in colloidal liquid materials. There's a scaling argument that the interfacial tension is like thermal energy scale kT divided by a molecular length scale squared, and if the molecules are large, macromolecular, then the surface tension is low. So that's interesting, that these interfacial tensions, surface tensions are low, but high enough to do these interesting things, pull on forces. There's also a lot of interest in the idea that there's self-assembly of other kinds happening at condensate interfaces. We have contributed a little bit to that literature. There's some really nice work showing that actually amyloid fibers can form at those interfaces, drive pathology. So I think the interface is, I'm tempted to make an analogy with the interfaces between fields, as you saw, in material science and cell biology is interesting. Any interface is very interesting. So yeah. Thanks for the question.
Thank you.
Very thought-provoking talk, really.
Yeah.
And because personally my field is rather towards the drug delivery aspect of life, and I was wondering if currently, do you think that the pharmaceutical companies, when they say that they're doing medicinal chemistry regimes, do you think there should be a paradigm shift between making small molecules interact with one protein, because this is the classical way of thinking of biology, protein Lego blocks, right? And shifting it towards a more organizational blocking kinds of medicinal chemistry.
Yeah.
How do you think this shift will happen? Do you see it shifting with your personal experience in your company as well?
Yeah, I think so. The question here is around if we're moving away from this kind of Legos or lock and key. I sometimes show a slide. Emil Fischer, 1893. That's the substrate enzyme lock and key picture, which that's basically defined our structural biology view and the idea that if I can insert something, put a pebble in there, then the key doesn't go in anymore. Now I have a drug. So with this paradigm changing around conformational heterogeneity and emergent structural ensembles, what is a path forward? And so these are very active discussions. It's clear that intrinsically disordered regions and these dynamic transient interactions that give rise to emergent organization would be wonderful drug targets if we could figure out how to target them, but it's going to be different probably. And I think maybe this is what you were getting at. I think it's probably true, that the action is going to be more in biologics, larger kinds of things than traditional small molecules. But I would say the jury is out. There's a lot of activity in the space. I think that some of what's happening, our company Laureate Therapeutics is using small molecule targeting strategies. But I think that primarily those are going to be useful for hitting the folded domains that are coupled to disordered regions. So for example, the oligomerization domains, et cetera. So yeah, but it's a very interesting question. I do think, if we fast forward however much time you want and say what's happened to change the way we do medicine, I think there has to be a new set of approaches. A lot of it is delivery too. If I get biologics, I mean, now I'm like, okay, how do I get it to the right place? So a whole set of issues, as you know. Yeah.
Thank you very much.
Yeah.
Thank you very much for your fascinating talk. My name is Yosuke Takahashi. I'm a neurologist in Kyoto University. So I'm very much interested in the pathogenesis of neurodegenerative diseases.
Yes. Yeah.
And probably the regulation of LPS formation would be a good way to intervene these intractable diseases. But problem would be if generally suppress the formation of LPS, it causes problems.
Yeah.
So if you want to treat, for example, Alzheimer's disease or Parkinson's disease, you should specifically suppress some kind of LPS. The other ones leave intact.
Yeah.
Do you have any strategy to specifically suppress the formation of LPS formed by, for example, alpha-synuclein or tau proteins?
Yeah. Well, this is challenging. I mean, it's the same problem, of course, in cancer. You want to kill the cancer cell, but not the regular cell, and all kinds of side effects, chemotherapy and radiation, et cetera. So same story. How can you be more specific there? I think it's tricky. I mean, the fundamental problem is that I'll make some blanket statements that are partially true, at least, that the molecular interactions that underlie these diseases are, by and large, I'll say, the same interactions that are, keeping cells, physiological processes. I mean, it's just that, you turn the knob a little too much, or there's, familial ALS, only 10% of ALS, right? But the knob is turned a little too much, and then you have. So I don't know. It's a very hard question. How do we specifically target, condensates that may be, setting up for pathological aggregation states? And I don't know. I mean, you would hope maybe there could be delivery strategies, or there could be strategies where you're only partially messing with the physiological, state, so there's not all these side effects. But this is a tricky business, yeah. In the same way that I would say cancer, is tricky.
Thank you very much. And may I ask you a second question?
Sure, yeah.
So if we cannot control the formation of LPS specifically, maybe we can treat the disease by inhibiting the Zoll-Gel transition.
Can you say it again? The
Zoll-Gel transition.
Oh, okay.
Yeah.
That's right.
So if the LPS can't go to solidify,
yes,
it will be beneficial for treating diseases.
Yeah, that's right.
Is there any good strategy to inhibit Zoll-Gel transition in LPS?
Well, there's a lot of interest in this space. I mean, there's a big, we were talking about funding in the U.S. There's actually a big federal program, this ARPA-H, to understand disordered regions and ultimately, the goal of treating devastating neurodegenerative diseases. I mean, I think that these sort of zipper motifs that are, I think that there seems to be some separation, and we were talking with some students yesterday about the residues or the grammar in disordered regions that are driving, dynamic, phase-separated condensates versus the residues that are more irreversibly zippering things together. I think that certainly is a place to look, in sort of these subsequent transitions or solidification or hardening that I do think are at the heart of many of the diseases that we want to target. So, I hope there's progress there. I think there's more questions than answers, but from the perspective of a doctor, that's bad, but from a scientist, that's interesting. So, yeah.
Thank you very much.
Thank you.
Hi. Thank you so much for such a incredible talk. I was very impressed.
Thank you.
I'm working on gene regulation by transcription factors. So my question is kind of related to the previous question. So as you know, the enhancer and the promoter making the sometimes the super enhancer, which is regulated by phase separation. So my question is what is the specificity of such kinds of things? Because there's so many active enhancer, but the target gene is always interact with the specific enhancer, and that the size of the droplet is also always regulated in some spaces, and it's super enhancer is easy to disappear.
Right.
When it's unnecessary. So what do you think about that thing, specificity and the regulation of the size, and why their droplets or condensate is easily disappeared in some stimulation or something?
Yeah, I mean, this is also, I mean, I'll just say for other people in the audience, as you know, that this area is, there's intense interest in what are typically called transcriptional condensates. I usually clarify because there's Pol 1 and 3, and that's people think of that's different. But yeah, the Pol 2 transcriptional condensates and super enhancers, and the work of Rick Young and Ibrahim Sissa, Arup Chakraborty and Phil Sharp and others. Intense interest in that space, and I think that I can just say what's challenging about getting at that, and you alluded to this as well in the size, is that these are very small. This is now we're at double, maybe triple digit nanometer sizes, so it's very difficult to understand and interrogate that level of emergent mesoscale organization. I think that's also what makes it interesting, but it makes it hard. There used to be more controversy in the space. There were these different camps, and the work of Bob Tejan and others. I think that there's a lot of convergence now on this idea, and they sort of talk about it in different ways, but it's the same idea of kind of this emergent nanoscale condensate. On the question of specificity, I think this is also gets back to some of the earlier discussion. Traditionally, the traditional paradigm, I would say, from sort of textbook structural biology pictures, specificity is dictated by shape complementarity and charge and all the things in a folded, protein, let's say, paradigm. I think that within this idea of conformational heterogeneity, disordered regions that are flopping around and transiently assuming folds, et cetera, you can get specificity, I think, there as well, but it's something different. It's sort of this emergent specificity, and we've worked on this a bit with Sigal Kadosh at the Dana-Farber Cancer Institute. We worked on these CBAF chromatin remodeling complexes, and it's the same. I'll say same old story. You have disordered regions in the structures that have been solved. You don't see them because people just there's nothing to see. But many mutations are found in those that are actually driving different cancers. So we've got to figure out what's happening. And it turns out there's specificity of interactions mediated by what a lot of people refer to as the grammar or the particular sequence of amino acids in those disordered chains. So that's, I would say, also very much that pushes against I'll call it a century of structural biology, right? So that's a hard one to make sense of, but it's very clear that we need to do that because, again, there's mutations in these things that are not structured in the traditional way. So somehow there's specificity or information encoded there. But yeah, I mean, I could just speculate and authoritatively wave my hands, as they say, but it's a very interesting topic, I'll say.
Thank you.
Yeah, thank you.
Okay, so maybe due to sort of time, this is the last question.
So first, thank you for a very interesting presentation, and I have one question. This question could be a little bit self-explanatory, but I feel like in the first part of your presentation, I think you have shown us about the concepts of valency and how it affects condensation.
Yeah.
And do you think mixing condensate component with different valency would modifies fluidity and structure of condensate? And is there a biomolecular condensate in actual cells that employs this kind of principles?
Yeah. Yeah, so I think the question is, you know, in the beginning of the talk, I outlined this idea of multivalency, and multivalency is, you know, an important factor in phase separation, and I got into the entropic driving forces and things like that. I think your question is very interesting. You know, are there molecules of different valence that, you know, would they mix, and how does that work, or do cells turn those knobs to, you know, generate structures? You know, I can just say there's a lot of work. We've worked a lot on that question, and you know, there's a paper, I think 2021, we did a paper in Cell. Sanders is the first author you could check out on stress granules and P-bodies and whether they mix or don't, and we think it has to do with exactly this question, you know, connectivity and valence of the interacting molecules. And this, we had kind of a, you know, graph kind of theoretic framework there. And similarly, we've tried to ask those sorts of questions as a, you know, more physical chemistry type of work. There was a paper, Rana, R-A-N-A is the first author. You could check that out. It's a Nature Chemistry paper from a couple of years ago on this idea of asymmetric oligomerization, you know, where you get actually different behaviors depending on the interaction strengths. I think that's, you know, so there's a lot to talk about there. I think cells are using those kinds of effects to structure multi-phase condensates. And there's quite some interest in harnessing that for forward engineering in applications to make synthetic condensates. For example, metabolic engineering. Yeah.
Thank you very much.
Thank you.
Okay, so thank you very much, Professor Brangwynne. So we really thank him again for his really wonderful and exciting talk.
Thank you.